This video will introduce transcription and cover the basics of this process. Transcription is part of a larger process referred to as the central dogma of genetics. In the most basic sense, the central dogma of genetics is the process of going from DNA, which largely just sits there, to proteins, which are the workhorses of our cells. Transcription is part of this larger process and refers specifically to making RNA from DNA. Hearing the name transcription can give you a sense of what it is doing. We are simply transcribing a piece of DNA into RNA language, which, if you recall, is remarkably similar to DNA. So transcription makes a copy of a piece of DNA here, but in the process does not alter the DNA in any way, thus preserving our genetic material in its original form. This ability to copy but not alter our DNA is key to reproduction and multicellularity. Within the process of transcription, not all bases are created equal. Specific sequences signal the transcription enzymes where to start and end RNA synthesis. When we refer to the process of gene expression broadly, we are talking about the creation of a functional product from a gene. Usually, this is a protein, and these proteins, along with our environment, go on to determine our traits or phenotypes. If we dig in to those specific sequences that signal different parts of transcription, we see the following. Regulatory sequences, which occur upstream from the promoter region, influence the rate of transcription. Often, these bind specific proteins that can either enhance or reduce the rate of transcription. The promoter site is a specific and highly conserved site where RNA polymerase, our main workhorse enzyme for transcription, binds and begins transcription. The terminator sequences signal our RNA polymerase molecule to cease RNA synthesis once they are reached. A final specific sequence, which is usually added after initial transcription, is the ribosome binding site. This will be important for the progression of our RNA to the next step in the central dogma, translation. In transcription, only one DNA strand is copied. This departs from what we previously saw in DNA replication, of course. Since only a single strand is copied, we need to pay attention to which one is being copied in order to accurately predict the RNA sequence. The names for the strand that is being copied are the antisense strand or the template strand. This strand is complementary to the RNA molecule being created and also runs anti-parallel to it. The sense strand or coding strand is identical to the RNA transcript with the exception of the thymine and uracil swap. An important note is that RNA synthesis, like DNA synthesis, occurs in the 5' to 3' direction. Let's conclude this video with an overview of the 3 stages of transcription. The first is initiation. In this step, transcription factors recognize and bind to the promoter region, enabling RNA polymerase to bind. This process denatures hydrogen bonds between our DNA bases in this specific region and thus opens up the helix for transcription to begin. The next step is elongation. This one is straightforward. RNA polymerase, in a processive way, moves down the DNA helix, synthesizing and ever elongating an RNA molecule. Eventually, this enzyme complex hits a sequence called a terminator sequence, which causes the whole transcription apparatus to dissociate from the DNA, separating the components. From here, the newly synthesized RNA molecule can proceed into post-transcriptional processing so it is ready for translation. We will dig further into each of these steps in the next video.